The overall goal of this experiment is to quantify the abundance and charging levels of transfer RNAs in Escherichia coli.
This method can help answer key questions in cell biology, such as how tRNA charging levels changes at different growth states. The main advantage of this method is that RNA levels can be quantitatively compared even between growth states that drastically alter the composition of the RNA in the cells.
Start this experiment by growing experimental and spike-in E. coli cultures according to the text protocol. Grow the spike-in culture at 37 degrees Celsius while shaking at 160 RPM. When the spike-in culture has reached an OD436 of about 0.1, induce the expression of tRNA cell C by adding IPTG. Grow the culture until it reaches an OD436 of about 0.5. Then, move the culture to ice until all samples have been harvested. After that, pre-warm one 100 ml capped culture flask containing 4 ml of 10% TCA for each sample in a 37 degrees Celsius water bath. Measure and record the OD436 of the experimental cultures directly before cell harvest. Then, transfer 4 ml of culture to a pre-warmed TCA-containing flask. Mix the sample by hand swirling the flask for 5 seconds, which will immediately disable all enzymatic activities, preserving the charging levels of the tRNAs. When all samples have been collected, add 5% spike-in cells to each sample.